thank you very much for the invitation and also for the fellow to participate here, to come from Uruguay, which is far away. So first of all, I just want to introduce the Advanced Bioimaging Unit and what we did and where we are. We are a joint unit, as Rocco said, in Hospital de Clínica and Institut Pasteur Montevideo. So we work with basic research in biology, but also translational research in the hospital. And we have like 4 parts in the unit. We do service, microscopy training, bioimage analysis, sample preparation, bioimage experiment preparation. Then we have a training and dissemination part. We conduct some workshops and training and lectures, seminars. We have grants to do that, so we bring people from around Latin America to participate with us. Then we do technology research. We develop some microscope mainly in FLIM, lifetime imaging microscopy, and also in spectral microscopy. And finally, we do research. We have our own line of research, developing biosensors, working with spectral pathology, cellular water dynamics, and so on. And we also have collaborations guided by research. So this talk I'm going to split into parts. First, I'm going to present the Phasor approach very quickly, how we use it, and then I'm going to present the library, which is the project I am working on, and the library we are developing at the unit. So the Phasor approach, it's a kind of mathematical analysis that we mainly use in these 2 kinds of signal, which is fluorescent lifetime imaging and hyperspectral imaging. The FLIM, it's basically you measure the decay of a fluorophore in a sample when you excite it, and the same for the spectrum. You measure the emission spectrum of the sample. And I'm going to somehow compare those characteristics with cofocal intensity, and try to not convince you, but show you why we use this instead of using normal intensity image. So basically, for FLIM, it's a characteristic property of a fluorophore. It is independent of the concentration of the fluorophore in the sample, but it's also changed with other factors like ions concentration, pH, hydrophobic properties, oxygen concentration, molecular blinding, molecular interactions, and that is useful to study different kinds of biological activities. For example, some dyes have the same intensity spectrum, but have a different lifetime. They are excited in the same wavelength, but they have a different decay, so you can differentiate them. And it's extremely useful in vivo imaging. For example, in this image here, you can study the membrane dynamic with Nile red, which change its decay depending on the water interaction. For the fluorescent spectrum, it's also a characteristic of the fluorophore. It can be used for unmixing without bothering the cross-channel. I'm going to talk about that later. Dyes have the same fluorescent excitation, but may have different spectrum. It's extremely useful to handle autofluorescence. I'm going to talk that because I do research on that, and solvatochromic proofs. And finally, something that we consider it's very important, it's nowadays most of the technologies have spectral detectors in confocal microscope, so it's very accessible in any facilities. So first, for FLIM, some important things to say is each pixel can have multiple species, and decays sometimes are complicated because you have like cross-bleeding or mixture decays in the same region. Biological samples have a limit exposure time because you may probably bleach them. And measuring that, you can do it very quickly and obtain an information from the sample. Fitting a resolution component at each pixel can be time-consuming, and interpretation is complex if you don't have like a mixing or something to get a simple image from that, because basically what you obtain is a stack. For spectral, it has some similar problems. Unmixing tools are great for splitting from fluorescent molecules marker. I mean, you can do unmixing with linear unmixing, but only if you know what you have in your sample. You have to know a priori that. Proposing models require expertise and understanding of the problem. I mean, biologically, you have to have an idea of what is going on in the sample to apply that mixing, to have the models to that. Fitting and resolution component at each pixel can be also time-consuming and computational consuming. And finally, you have to trust in commercial softwares to do that. For example, size in the software, they have the linear mixing. You just select the spectrum, but it is theoretically embedded there, right? So a little of the story of the Phasor plot, it was defined by Gregorio Weber in 1981 and later introduced mathematically by David Jameson for frequency domain and time domain in FLIM. And the advantage they show that it has over intensity, you don't, or other methods, you don't need knowledge a priori. It has some simple rules to understand that I will forward them, and you don't need fitting. I mean, you don't need a model to do the fitting. So basically, what does the biologist do when they do Phasor plot or Phasor analysis? For example, in the case of a spectral imaging, they take a spectral image like this one, where you have a spectrum in each pixel in the lambda domain. They apply the Phasor transform. The Phasor transform is based on the Fourier transform. So basically, you go from this space, the intensity space, to a complex space, and you obtain G and S coordinates, which are basically X and Y in the axis. Okay, so for each spectrum, you will get like a dot in the cluster that it is unique by the Fourier transform properties. And basically what they do is to look for component, spectral component or lifetime component into the cluster that will represent or belongs to different regions in the sample. It has some properties that are very useful. I mean, visualizing when people do the analysis is that the change in the phase corresponds to a shift in the spectrum. Also, the modulation, if the spectrum is more wide, the radius from the original point will be smaller. And finally, since it's a linear transformation, the transformation, if you have a combination of two pure spectrum, like here, the red one, it will rely on the line between the two pure components. So now I'm very quickly, I'm just going to talk about two applications we have or you can do to do analysis with Phasor plots. It's spectral mixing with phasors. This was developed by Lionel and Alex Badmijana. Basically, what you want to do is unmix what you have in a pixel, right? To take the fraction of each component. If you have two pure components, it's very easy because you know that the combination relies here, and it's basically the fraction is this distance or this one. You also can write it as a linear equation. But if you have more components, I mean, you can theoretically resolve mathematically using the Phasor transformation for many harmonics. So this way you will be able to unmix many components theoretically, not experimentally. And how it looks, for example, in this paper, they just stain a sample with 5 components. Those components, ideally, in a perfect world, would stain each organelle, right? Like DAPI for nucleus, mitotracker for mitochondria, for example. But in fact, what you will have bleeding, so you won't have the pure stain in each pixel or in each organelle. So the bleeding, you can see it in the spread of the phasor. So applying the spectral mixing, you can unmix and reveal the final image, which with the component of in each pixel for each spectrum. And the other application I want to talk to you about is the Phasor plot in autofluorescence. It's really useful in autofluorescence. This was my thesis in the undergrad. Basically, if you have this is just a pathology image that the dermopathology used to differentiate nevus and melanomas. I'm not going to get into this, but basically the melanoma is the malignant tumor, and the nevus is the benignant. And what they do is just to analyze. There are some AI algorithms, but the final decision, they do it looking at the image. So we propose to use spectral imaging and Phasor analysis and see if we can see here where the tumor is and here where the tumor is, there are spectral shifting. So what we show is that I don't know about pathology, but what I know is that the spectrum components are very different in the region of both tumors. So I can create a pseudocolor image which differentiate the regions, right? And that's like the power in autofluorescence. Then there is a bigger study there that we did, but then if we know which component we have on the skin with other methods, we can start to study the distribution over the phasor and the spectral distribution and create image of this region. I'm going to talk about the PhasoPy library now. A little of a story, Enrico Graton, in the last 40 years, he developed this software to do all of this kind of analysis on his own. He did the development and the maintaining. Well, he retired, so we lost everything. And that's what motivated to create PhasoPy. PhasoPy relies on that. We do an image analysis, simulation acquisition, film, FRED, GP, and spectral phasor, not the rest. And the other part was, while I was doing the autofluorescent analysis, I need to develop things in Python because I couldn't use this software for huge image. So I developed HSAPy, which is a very, very simple library in Python. And with that, we apply for the grant. We get the grant in the 5 cycle for the open source software. And we have like two parts, develop the library and create a community around that, because there are people using this, but everyone is like spread. So for the community part, I'm going to show now the technical details of the library, but for the community part, we want it to be, well, open, right? Collaborative, permissive, inclusive, integrate with the Python ecosystem, have support and disseminate it. The current team, it's this, Lionel is leading, Bruno and I, we are developing, and Christoph is leading the project infrastructure, and Michelle, as the PI, took over Enrico when he retired. The project infrastructure, what we have is, well, the web page for the library. It has a GitHub organization, a repository, a Zenodo community where we deposit all the data. We are creating data for that, and we have some companies that have provided data to us. Well, it has a Python package index. Documentation, it's very important for us to create a strong documentation with tutorial, AIP reference, contribution guides, and code of conduct. Well, contributions are very welcome. So we have a contributing guide, questions, suggestion, bar report. We want to share data files. I mean, if you want to propose something, it has to be open, and you have to open the data for that. Requirements, the requirement to use the library, I basically know the Phasor approach, a little of image processing, array programming, Python. It's for Windows, Linux, and Mac. It's based on Cython, actually, and some libraries. All the part of the reading files came from Christoph libraries. And it looks something like this if you enter in the web page. This is the AIP reference. The tutorials for each module we are developing, we have the tutorials with examples and results. So right now we develop the Phasor plot file readers. We have all the file readers. We created the functions to perform to work with hyperspectral time domain and frequency domain signals, lifetime and FRED, apparent and lifetime, a single lifetime, polar coordinate for the frequency part, and right now we are working on the calibration, threshold, filtering, and mixing part, because actually, since we want to make it a little, I mean, very independent from other libraries, we are developing all the, I mean, we develop median filter, wavelet filters, the FFT algorithms. We use the NumPy algorithm, but we also develop our own. Now the dissemination of the project, because when we write the grant, I mean, and the idea of creating the community around PhasoPy and Phasor analysis in film and spectral imaging, it's it's not just about developing the code and putting open somewhere and people expecting people to download it and use it. So we have other funding to develop some workshops, mainly more for microscopy itself, not image analysis. But in that workshop, one is the workshop in advanced microscopy and biophotonics, where we do film and spectral imaging. We do the acquisition. I mean, we have like theoretical lectures, and then acquisition, and later image analysis. But the image analysis there was done with SimFCS, the software that I mentioned. It was a I mean, now Enrico opened the license the last years. It doesn't have more support. So it's like shutting down. And in the workshop, we use that. But when we apply for this grant, we say, well, part of the idea is to disseminate what we are doing. And to do that, we introduce some practical session in the workshop with very simple material, mainly based on Jupyter or in Google Colab, depending on where we did it. For example, in the Institut Pasteur, we have a computer lab, so we can install everything that we want. But a couple of weeks, I went to California with Christoph Golk. We did the practical session there, so it would be easier since every student has their own computer to work on the Google Colab. So in this part, the library is very, very new. Actually, last month, we released the first version. So we can like carry on an entire workshop on that. But we've been introducing it because the majority of the people that came to this kind of workshop, they are already working with spectral imaging or film. And what I saw is that the majority of them, they have someone programming or collaborate with some programmers or image analysis to ask for them to create, mainly in Python, right, the code to do this kind of process. So the idea of creating the community was, well, it's an open source library, it's in Python. The idea when we write the grant is to create something like scikit-image or NumPy. So please come, what you are doing, like put it in our repository with our contribution guidelines. If you are a user, I mean, if you need to use these codes, write to us, we can help you, or because not everybody has someone that program in their labs. So what we are doing now is, in this kind of instance, we are trying to reach these people. I mean, the ones that are like users, because they do experimental things and they need the code and these things for their data. But also, like in the case of Marcelo Socoler, that he works for people that need this, and he's developing things. And in the case of with Marcelo, what I want to mention is that when we got that grant, we also got another grant to develop an Apari plugin. It was my first time working with a plugin, and I really didn't know how to work with that. But Marcelo really, I mean, he taught me everything and how to do it. But at that time, we didn't have a PhasoPy, so we have to develop all the code and do it like a little acceptable to put it into the plugin. It took a lot of time, and Marcelo actually was developing the same thing, but for Flim. And now we team up, and we are doing like Napari Phasors, which encompasses everything together, and it's more sustainable and self-sustainable because it's based on PhasoPy. And I just want to transmit like this idea that we take advantage of other grants that we have for training, and we introduce the library there that we are developing because it is related to that. And somehow all of the project we have, I mean the founded project we have in our unit, they are independent, but they work together, and this way we can achieve something like further. Because, for example, some people that participate in this workshop, I mean, they are not extremely trained on PhasoPy or in programming because it's just a practical session from 3-4 hours. But then they came back to their communities and they start to contact us to work with this. And that's the way I have found like better to following up the use of the library right now that is very, very new.